you all for coming today and for your attention to this talk. I'm Dr. Brian Walitt, and I'll be presenting some data from the NIH's intramural study today. As Vicki had mentioned, around 2015, the National Institutes of Health refocused its efforts to study chronic fatigue syndrome. Part of this was an extramural effort that was led by Vicki Whitmore, and part of this was an intramural effort that was led by Dr. Avindra Nath, which I was part of. The overall structure of the intramural was to develop a protocol to study chronic fatigue syndrome, myalgic encephalomyelitis at the NIH. And the hypothesis was that post-infectious ME/CFS is triggered by an infectious illness that results in immune-mediated brain dysfunction. And to study this, we developed a cross-sectional study to deeply phenotype individuals with PI ME/CFS to define its pathophysiology. That was started in 2015, and the fruits of all of that labor was published in February of this year as this paper in Nature Communications. There were 75 authors that were part of this project, different doctors and scientists. 15 of the 27 institutes at NIH contributed to this work. There is a lot of data and a lot of information that was part of all of this project, and we put on a symposia to present this material to the world. It's about 5.5 hours long, and it's on the internet, and this QR code would bring it to you. I put it up here now because I'm going to give a very quick overview of the main results, but everything that I talk about is talked about in more detail by the scientists that did the work as part of the symposium. So the study was designed as follows. Participants came for an initial visit at the NIH Clinical Center for a deep phenotyping, where they had a detailed clinical evaluation, collection of physiological measures, and biological samples. This was about a week-long stay at the NIH. Once that information was collected, it was put together and sent to an adjudication group that reviewed all the cases and had to unanimously agree that the cases were both post-infectious and ME/CFS. And those participants that were adjudicated to be appropriate participants were brought back for an exercise stress visit. These are the deep phenotyping measurements and the different individuals that contributed to all of the different measurements that were performed. As you can see, some of these were clinical measurements, neuropsychological measurements, neuroimaging, and physiological measurements, and a lot of different types of biosampling. For our case adjudication process, all of that information we collected during our week-long stay was summarized into briefings that were sent to a number of expert adjudicators in the community. The panel independently reviewed each of the briefings and voted about whether they thought the cases were appropriate, and only cases that were unanimously agreed upon were allowed to be used in our study. And I'd like to thank our adjudicators, which were Lucinda Bateman, Andy Kogolnik, Anthony Komaroff, Benjamin Nadelson, Dan Peterson, and Avi Nath. Participants also came back after that for an exercise stress measurement where we used a one-day CPET paradigm to try to understand the consequences of PEM biologically. This is the focus of some ongoing analysis currently. In terms of recruitment, we talked to around 500 people who wanted to be part of our study, and many of them, for one reason or another, were not eligible to participate because of our tight inclusion and exclusion criteria. After all of the work was done, we brought 25 healthy volunteers and 27 ME/CFS participants to campus, of which 21 healthy volunteers were actually healthy, and of which only 17 of the ME/CFS participants actually met and went through were adjudicated as appropriate participants. There would have been more participants in the study. However, the SARS-CoV-2 pandemic occurred, and that closed down our operations for 2 years, and so we moved forward with our analysis. When you look at the groups here, what I'll be doing is presenting the healthy volunteers in blue and the ME/CFS participants in red throughout the talk. What you can see is they were pretty well matched in terms of age, sex, race, ethnicity, and education. On average, participants with ME/CFS had it for about 33 months, and all of them met the IOM criteria. In terms of symptom reporting, this sort of gives you a sense of all the different ways that we measured individuals in terms of their complaints. And you can see that they are vastly different from the healthy volunteers. And on the right are some graphs here to show you sort of the distributions, the healthy volunteers and the ME/CFS participants. In terms of measurements of fatigue, there's an immense gulf. In terms of overall symptomology, you can see that there is a pretty big difference. But as you get to sleep, that difference is still there, but less, even less with like attention. And in terms of anxiety, there's a difference, but you can see how small that difference is. Overall, the work led to us putting together this overall hypothesis of how an infection can be transformed into ME/CFS. And I'm going to go through each of these little pieces quickly for you. One of the major issues here that I'd like to bring into your attention is that part of these appear to be reactionary, that you have an infection and it drives a process forward to the brain. But once it gets to the brain, there seems to be a series of vicious loops that work together that may be a self-perpetuating phenomenon. And that these things may all work together to lead to the development of the PI-ME/CFS phenotype. And first, let me start here with the reactionary pieces. And so one of the most important observations is that PI-ME/CFS has disease-specific immune signatures. What I'm showing you here is some data from our flow cytometry experiments that we did on blood and cerebral spinal fluid, where we were able to show that there were altered B-cell phenotypes that suggest that the B cells are not maturing in the way that they're supposed to. That suggests that they sort of get stuck in an immature state. We also found that there are alterations in T-cell markers that suggest T-cell exhaustion and activation, in particularly PD-1 in cerebral spinal fluid cells, and CD226 being lower in CD8 positive T cells in the blood. Further, that these differences, when you further look, were actually also sex specific, that each men and women have different immunological issues. Further, the gut microbiome in ME/CFS is different. In terms of our analysis of stool samples, we were able to show that the microbiome of PI-ME/CFS participants was less diverse and had different proportions of microbial species. And further, when we actually looked at the species that were different, many of them had been previously reported in the literature, and so we were able to confirm prior findings. And a number of those particular species are butyrate-producing bacteria. Butyrate is an interesting compound. It has a number of important effects on human physiology, but we are unable to produce it on our own. We rely on bacteria in our gut to ferment it and to absorb it, and then that's how we get our butyrate. And the idea that there's a difference in butyrate bacteria is an interesting observation, and you'll see why in a little bit. Further, birth sex is biologically important in understanding PI-ME/CFS. I'm showing you gene expression data from immune cells and muscle cells here. And what you can see is when you look at patients and controls, you can't really differentiate using traditional analyses. However, when you segregate into men and to women, suddenly the groups segregate nicely. And you can see this is for the PBMC RNA sequencing, and you see the same kind of phenomenon when you look at muscle RNA sequencing. In almost every biospecimen that we looked at, we see this relationship, whether it be proteomics of the blood, proteomics of the cerebral spinal fluid, lipids, metabolomics of the cerebral spinal fluid, that there is a profound biological difference in ME/CFS that is gender-based, that needs to be considered going forward. However, cerebral spinal fluid metabolomics was the only biocompartment that was able to discriminate PI-ME/CFS when we didn't consider gender. And you can see here in our most rough analysis, you get some separation, But when we consider all participants using differential expression analysis, you can really separate the groups on their own. And this is it suggests to me that the cerebrospinal fluid metabolomics might be the best potential biomarker for looking at ME/CFS. What was different in these groups? Decreases in glutamate, dopamine 3-O-sulfate, butyrates, polyamines, and TCA pathway metabolites. All things that have come up in the literature before. Further, there were other alterations in the CNS, the cerebrospinal fluid. When we looked at neurotransmitters, there was decreased metabolites of catechols. DOPA and DOPAC are metabolites of dopamine. DHPG is a metabolite of norepinephrine. And all of these were decreased in PI-ME/CFS participants and seem to be a potential way that we get to autonomic dysfunction in these individuals. So from here, we take these decreases in CNS metabolites into the brain, and we see alterations in brain function. And let me go through some of that now. We developed a grip paradigm based on this very simple and easy to repeat observation that people with PI-ME/CFS are able to grip the same in terms of maximum strength as healthy volunteers, but their ability to maintain their grip falls off precipitously. And so we tried to understand where that is coming from. And so we looked at it in a number of different ways. This is a grip, MRI safe grip device. And we did a paradigm where we had individuals grip for 30 seconds, rest, grip, rest, grip, rest. And we made all sorts of physiological measurements of the entire motor circuit. We measured how the muscle was working using electromyograms. We measured how the motor cortex was functioning using transcranial magnetic stimulation and EEG measurements. And we also repeated this experiment in the functional MRI environment to understand brain activation. And so what we learned is that in terms of the change in grip, right, we used a measure of lactic acid decrease that you can get with EMG called the Dimitrov index. And while from the last good block to the beginning of fatigue in healthy volunteers, you see an increase in this index that suggests increases of lactic acid. You see decreases of this in the ME/CFS participants, which suggests that it isn't peripheral muscle fatigue that is the cause of this early drop-off of activity. When we looked at how the motor cortex was functioning, we looked at motor evoked potentials. In healthy individuals, as people get more tired and use the resources of their motor cortex, the ability to have motor potentials decreases. But in the ME/CFS participants, it increased over time as failure happened. And this is suggestive that there is not a decrease in the motor reserve of the motor cortex in these individuals, and that neither peripheral fatigue or central fatigue are the cause of the early failure in grip strength. However, when we repeated this again in the functional MRI environment, what we saw is that there was no difference in how the motor system was working, but there was this area of the brain called the right temporal parietal junction that was activating differentially between the groups over the course of the task. Here you can see in the healthy volunteers that area increases and increases as they develop fatigue, and in the PI-ME/CFS participants, you see this sort of decrease in the function over time. The TPJ is responsible for high-order integration of brain function and serves as a mismatch detector. And to us, this suggests that there is a defect in neural circuit integration that is interfering with the ability to trigger the motor system appropriately. That's not the only thing that's happening, right? At the same time, we believe that there's the same chemicals are having secondary effects on the autonomic nervous system and cardiorespiratory function, that you have more than one consequence from the same alterations. One thing that we were able to demonstrate is that there is autonomic dysfunction in our PI-ME/CFS participants. While individuals were placed on a tilt table and abnormal responses were common in both groups, we were able to show that heart rate variability measurements were very different between the two groups, showing that there's decreased parasympathetic activity in the PI-ME-CFS participants. We also performed a one-day CPET on individuals. This gives you a sense of the bike and the ramp that we used. Individuals cycled about 8 to 10 minutes. All of them had an adequate cycle response. All of them met their RER of 1.1 for those playing at home. And what did we find? What we found is that, as the literature has previously reported, that people with PI-ME-CFS hit their anaerobic threshold earlier than the healthy volunteers. And not just earlier, but so early that some of the individuals were in an anaerobic state, even with less activity than just walking downstairs, right? And then so some of this impairment seems clearly to be to the speed at which they run out of their aerobic potential. Further, what we found is that there was an alteration in heart rate responses with exercise. You can predict a heart rate response with exercise using a very simple equation in exercise science, and our healthy volunteers follow that almost perfectly. However, our PI-ME-CFS participants, they are not able to mount the required heart rate response to really meet the metabolic demands of exercise. This is known as chronotropic incompetence, and is usually thought to be a phenomenon of dysautonomia. And so this is yet another way to show autonomic dysfunction. Further, we placed individuals in this metabolic chamber, an indirect calorimeter that measures total body energy use before and after exercise. And what we found is that there was no differences in total body energy use before or after. Right? And so it seems that the bulk of the deficit is occurring with activity, not with at rest. And so, as you can see, all of these different arms and pathways have the ability to decrease our ability to do things, right? Whether it interferes with our ability to start movement, maintain movement, or push our activity. And this all leads to a reduction in what we're able to do with PI-ME-CFS and contributes to the PI-ME-CFS phenotype, right? You don't start here, but the infection pushes the process that leads to that reduction of activity and the development of the phenotype. So in summary, what we learned from our recruitment process is that many of the individuals have other underlying diseases. About a third of the participants that we evaluated, we found an alternative cause for PI-ME-CFS. And so understanding, deeply looking at patients and following them over time to understand what emerges is very important. We also had 4 of our participants, while we followed them, to actually spontaneously recover. And that does happen, and it's important to understand why that happens. Further, multiple biological systems are involved in PI-ME-CFS. It seems that the immune system and the microbiome may be catalysts to central nervous system changes that play a critical role in the maintenance of the phenotype, and that there are multiple targets for intervention, in particularly understanding the sex differences may lead to improved outcomes and improved for clinical trials down the road. Again, I'd like to thank everybody that was part of this project. Here are all of the 75 authors and all of the other people that contributed in various ways to this work, and my eternal gratitude to all of the PI-ME-CFS participants and healthy volunteers that were part of the project. And again, further information, including hearing directly from the participants themselves, can be seen here at this web link here for our symposium. Again, thank you so much for your time.